Here we're going to look more closely at the diagram that you should be doing to summarize all the steps that happen in cellular respiration. So the idea here is that you understand what are the reactants that are coming in into each step of cellular respiration, what are the products of each of those major steps, and how do those relate to the overall process. Let's look here at glycolysis, which is the beginning of cellular respiration, and glycolysis is the process that any cell can do. Any eukaryotic, prokaryotic cell is capable of doing glycolysis, and glycolysis happens in the cytoplasm, so regardless of whether you have mitochondria or not, any cell is capable of doing glycolysis. So glycolysis does not require oxygen. It's the only process that doesn't require oxygen, and is the process that, again, happens in the cytoplasm, so it also doesn't require mitochondria. So in your book, you can have a diagram like this showing every intermediate step, but we're not going to go into all that detail. Instead, all we're going to focus on is what goes into the process. So glycolysis starts with a six-carbon glucose molecule, and we're going to track the carbons in this glucose molecule. So those 6 carbons, where do they go? We're going to track net ATP production. So notice here you have 2 ATPs that are used and then 4 ATPs that are produced. So you have a net yield, a net gain of 2 ATPs. So that's all we're going to have here. And the other big thing that happens is you have electrons being transferred from the glucose molecule into electron carriers. So you have NAD plus being reduced as it gains a pair of electrons. It becomes NAD, you could say NAD minus, and then a proton binds to it, so now it is NADH. So this is, NADH is the form carrying a pair of electrons, and those electrons were harvested from the glucose molecule. And the end product, pyruvate. So the glucose molecule is now split into 2 pyruvate molecules, and each pyruvate molecule is 6 carbons. So we have here glycolysis, again, as summarized as we had it. So that's the first step in cellular respiration. Now the next decision the cell has to make is whether, if oxygen is available, aerobic respiration will continue. And aerobic respiration happens inside the mitochondrion. If oxygen is not available, then this will be the only step that takes place in the cytoplasm, and then you will have fermentation. That the main purpose of fermentation, as we're going to talk in another video, is to be able to restore this NAD+, because eventually the cell runs out of NAD+, and it can no longer continue glycolysis. So the main purpose of fermentation is to restore NADH in the absence of oxygen so that glycolysis can continue. But here we're going to say that oxygen is available, so there is oxygen in the cell. Therefore, the following steps will take place inside the mitochondria. And the next step is pyruvate oxidation. The name is easy because you start with pyruvate. So this is the oxidation of the pyruvate molecule. In that oxidation, you're removing a pair of electrons from each pyruvate. So you have a net of 2 pairs of electrons removed from pyruvate and given to NAD+, and that becomes NADH with those electrons. Another thing that happens is you have a 3-carbon pyruvate and you end up with a 2-carbon molecule. So a carbon from each pyruvate is now lost as CO2. And CO2 is a gas, and it's a gas that we breathe out. So in the process, we already lost 2 of the carbons in this original glucose molecule and have now converted them into CO2 that we simply breathe out. So this is important because the mass of this glucose molecule is still there, the carbons are still there, but now instead of being part of a glucose molecule that contributes to our weight, it's now part of a CO2 molecule that we just breathe out. So we're losing weight by breathing out this CO2 from this process. But it's not that the mass has changed, it simply has changed form from a glucose molecule into CO2 form. So the other thing that happens, okay, so we have 2 CO2s, in total 1 CO2 for each pyruvate. The other thing that happens is we have a coenzyme A, so this is just a cofactor, something necessary to continue the reaction on, but it doesn't get consumed. So it gets recycled. So we don't really take it into account in the carbon count because this will get recycled. But this molecule that has 2 carbons original from pyruvate is now called acetyl for the 2 carbon and CoA because it's bound now to this CoA. So this process is to get the molecule ready so that it can move on to the next step. And the next step is the Krebs cycle or the citric acid cycle. And in this in the Krebs cycle, you do produce 2 ATP molecules, one for each pyruvate. So think here that since we split the glucose molecule into 2 pyruvate molecules, 2 3-carbon molecules, everything from here on happens twice. And the numbers I have here are for the overall process. So the 2 ATPs, 1 ATP from each Krebs cycle that runs from the glucose molecule. So this is total for 1 glucose molecule. You also have 4 CO2 molecules that are being produced. So these 4 carbons that we ended up here are now completely turned into CO2. So the carbons of the original glucose molecule at this point have all become CO2. 2 that were converted here and 4 here. So at this point, at the end of the Krebs cycle, you have none of the weight of the glucose molecule in the body anymore. It's now turned into a gas that is carried in the blood to the lungs and then breathe it out. But the main thing of the Krebs cycle, although you do produce 2 ATPs, the main yield is in harvesting electrons. So in the overall process of glycolysis or cellular respiration is taking electrons from the glucose molecule and giving them to these electron carriers. Here we have 6 pairs of electrons that were harvested and were given to 6 NADH molecules. Again, this is for the overall. So there are 3 NADH produced for each Krebs cycle. So this is total per glucose molecule. And there is another electron carrier, this is FADH, which also takes a pair of electrons and becomes FADH2. This one FADH2 is produced for each Krebs cycle. Again, here are the totals for the entire glucose molecule. So like we said, this process requires oxygen. And up to here, if you look at the yield of ATP, we have the 2 ATPs that we produce, the net gain from glycolysis. And there were 2 ATPs that we produce from Krebs cycle, but those 2 ATPs are used to bring NADH into the mitochondrion. So at the end, you have no net gain. So we've done all this process, but so far we've only have a net gain of 2 ATPs. So why will the cell go through all this trouble if it cannot get any ATPs? And the thing is, the energy of the glucose molecule is mostly in these electrons that are carried by NADH. So the energy is here, these high energy electrons that were taken from the glucose molecule and were given to these electron carriers. And now the main step is getting the energy from those electrons and being able to use that energy to make ATP. And that process of getting the energy from the electrons that the electron carriers are carrying with them and using that energy to make ATP happens during the electron transport chain and chemiosmosis. And this process, the electron transport chain, is oxygen dependent. So it needs oxygen and can only happen in the presence of oxygen. So that's why all the processes inside the mitochondrion, so everything we have here inside the mitochondrion, all is oxygen dependent, because the main purpose of harvesting these electrons is to take them to the electron transport chain, which we're going to see next, and that requires oxygen. The electron transport chain is a set of 3 proteins, and these proteins are proton pumps. So you can think of them, they are proteins that do active transport, and the active transport that they do is move protons from the inside of the mitochondria. This is the mitochondrial matrix. They move protons from here, the mitochondrial matrix, into the intermembrane space. So this is the inner mitochondrial membrane. And then, as you know, the mitochondria has 2 membranes. So it has this inner membrane, and then you have the external membrane that is not drawn here, but is somewhere right next to this. So the protons are being concentrated in the intermembrane space, which is the space between the inner membrane and the outer membrane that should be right under here. You don't need to remember the names of these proteins, but you need to know there are three. The main one is called the NADH dehydrogenase, because this is the one that removes the pair of electrons from NADH, and also NADH is restored back to NAD+ once those electrons are removed. The BC1 complex and the cytochrome oxidase complex is the last protein in the transport chain. So NADH comes and it donates its electrons to the first protein in the electron transport chain. So the pair of electrons are given to the first protein, and NADH is restored back to NAD+. So this is very important because now NAD+ can go on and go back to glycolysis, or go back to pyruvate oxidation, or to Krebs cycle, which need this NAD+ in order to continue. So here the NADH is recycled back to NAD+, and the electrons are given to the first protein in the electron transport chain. Now think these electrons are very high energy electrons. These are high energy electrons that were taken from the glucose molecule. So you can think of them as like fully charged batteries at this point. And the energy in those electrons is used by this protein to actively transport a proton from the mitochondrial matrix into the intermembrane space. So this protons are just a hydrogen that is missing an electron. So a hydrogen atom is just an atom made of one proton and one electron. So once you take the electron, all you have left is the proton. So the energy here has been used to pump this proton against its gradient. So there's high concentration of protons here and low concentration in the matrix. Once the electrons are used by the first protein, they're given to, this is a transferring carrier protein that takes them to the next one, the next proton pump. Now the electrons have less energy than what they had originally because the energy was already used by the first protein. So they have a little bit less energy, but this protein is able to, this BC1 complex is able to use the energy in those electrons to pump another proton. So again, using active transport, it's moving a proton from low concentration here into high concentration in the intermembrane space. And finally, the electron is transferred to the last protein in the electron transport chain. So this protein squeezes out the last bit of energy it can from that electron, from that pair of electrons, and uses that energy to move electrons from the inside of the mitochondrial matrix, into the intermembrane space. So a third proton is being pumped into the intermembrane space, and now the electrons have all their energy that is usable by the cell has been used. So these electrons now become a problem because they no longer carry energy, so now they have no value to the cell, and the protein needs to get rid of them so that it can take new incoming electrons that have energy. And so these electrons need to be discarded. So think of it as, once you have old batteries in your remote control, you want to get rid of those dead batteries so that you can put in new ones. If you cannot take the batteries out, you won't be able to put new batteries in. And here is where oxygen becomes important. So oxygen is a highly electronegative atom, and it's craving, think of it as somebody who is eager to get electrons. So this oxygen wants to take those electrons in, and it will take it regardless of how low energy they have. It just wants to take those electrons. So the electrons are given to oxygen, and once oxygen gets those electrons, it combines with two protons and now forms a water molecule. So the oxygen that we breathe in, the purpose of it is to get the electrons at the end of the electron transport chain after the cell has taken all the energy it can from those electrons, and the electrons are given to oxygen, and oxygen forms now water with those electrons plus a pair of protons. So this is important because we think that the oxygen we breathe in comes out as CO2, but that's not true. The oxygen is used to take electrons and then form water. We said there is another electron carrier, and that is FADH2. Once it takes electrons, and FADH2 the electrons that it carries have a little bit less energy than those that are carried by NADH. So FADH, when it brings its electrons, it skips the first proton pump and it gives the electrons to the carrier that is going to take them to the second proton pump. What it means is that the electrons carried by FADH2 don't get to activate the first proton pump. They get transferred to the second pump, which will use the energy in those electrons to pump one proton against its concentration gradient. And then the electrons will go into the third or last proton in the electron transport chain, and the energy in those electrons will be used to pump a second proton. So only 2 protons get pumped by the electrons carried by FADH. So these only the last 2 proteins in the electron transport chain are activated by the electrons that FADH is carrying. And finally, those electrons, once all their energy is used, they're given to oxygen, and water is formed. So again, if we look at the overall process, the electrons have been moved from the electron carriers and are given to these 3 proteins in the electron transport chain, which use their energy to pump protons. And then once their energy is used, the electrons are given to oxygen and form water. But up to this point, we haven't produced any ATP. All the energy is now in this proton gradient of this highly concentrated protons in the intermembrane space that are not only they're all positively charged, so they repel each other, they want to get out of here, but they're also at very high concentrations. So there is an electrochemical gradient. So it's not just the high chemical concentration of the protons, but it's also they're having the same charge that is pushing them to get out of the intermembrane space as much as possible. This is where the cell takes advantage of that and finally is used to convert this energy that is in this gradient into ATP. So the protons that want to get out of the intermembrane space are given a way out by the cell by this protein called ATP synthase, or this enzyme. And this enzyme is kind of like a turbine, it's a rotor protein. As a proton comes through and it wants to get back into the mitochondrial matrix, it rotates ATP synthase, and ATP synthase uses that energy to combine an ADP with a phosphate and form ATP. So this proton gradient is now used, the energy in that proton gradient is used to make ATP. So using that electrochemical gradient as protons move through, making their way back into the inner mitochondrial matrix. Now, remember that NADH was able to activate all three proton pumps, so it was able to pump 3 protons against the concentration gradient, the electrons that NADH was carrying. On the other hand, the FADH, sorry, this is just one FADH. For each FADH2, you can pump 2 protons. So, assuming that every proton here makes its way through ATP synthase, you will be able to produce 3 ATPs by each NADH, and you will be able to produce 2 ATPs for each FADH2. So these are the maximum theoretical values because not every proton actually makes its way through ATP synthase. Some protons can sneak through the phospholipid bilayer and therefore they won't be used to make an ATP. So these values are not always achieved by the cell, but this could be the maximum possible. So doing the math, we produce 10 NADHs during the overall process. So we had 2 NADH producing glycolysis, 2 NADH produced by pyruvate oxidation, and 6 produced during Krebs cycle. So that will be a maximum of 30 protons being pumped, and that will mean 10 times 3, a maximum of 30 ATPs produced by NADH in total. Now FADH, we produce 2 FADHs, because we only produce them during Krebs cycle, 1 for each pyruvate. And if each FADH pumps 2 protons, that will be a total of 4 protons being pumped, which would give us a total of 4 ATPs being produced. So this is where the maximum numbers come from, a maximum of 30 ATPs produced by NADH, a maximum of 4 ATPs being produced by both FADH molecules, and the 2 ATPs that we produce during glycolysis, and that gives us the maximum possible value of eukaryotes of 36. But like we said, this is not always realized because not all the protons actually make their way through ATP synthase, so not every proton results in an ATP being produced. Still, the expected values are somewhere between 30 and 34 or 36.